Hello everyone. In this video I'm going to be talking about the transport of carbon dioxide. Now this video is aimed primarily at Key Stage 5 students, particularly those studying the OCR syllabus, where you look at how oxygen is essentially delivered to respiring cells, and then you conversely look at how carbon dioxide produced in those respiring cells then leaves the body essentially, and how you deal with that carbon dioxide. So what we're going to do is first of all just very quickly draw in a small picture here, just because then what we're going to do is label these parts. So we're going to have here some tissue cells. We'll draw in just the nuclei there. Then we're going to have, let's say, in the middle, this region here that I'm shading in blue. This region here is going to be our interstitial fluid. Then we're going to pretend that over here is a very large red blood cell. So we'll just shade the outside in red here. I'm not going to do the whole thing. My picture is still looking a little bit rough around the edges, but still. So let's just label what we have here. So we've got the tissue cells over to the left, we've got interstitial fluid in the middle, and then this here is our red blood cell, which would make this whole white region here the plasma, the liquid part of the blood. So now we know that carbon dioxide is produced in respiration, and for this we'll do this in green just to make it stand out a bit. So we're making carbon dioxide, CO2, in these respiring cells. Now some of that carbon dioxide passes from the cell through the interstitial fluid and dissolves straight into the plasma. Now there's three ways carbon dioxide travels, and this is the first way we're going to look at, and actually 5% of all of carbon dioxide produced in respiring cells dissolves into the plasma. So we have CO2 dissolved in the plasma, and like I said, we're looking there at around about 5%. So that explains what happens there. Now, we also have carbon dioxide associating with hemoglobin in the red blood cell. Now, the red blood cell I'll sometimes refer to as an erythrocyte. That's its scientific name. So some of the carbon dioxide produced in the respiring cells actually binds with hemoglobin in the red blood cell to form what's called carbaminohaemoglobin. So I'll just say that again, carbaminohaemoglobin. And this process is actually reversed in the lungs. So I think we'll just use another colour to show a slightly different one here. So this CO2 actually comes into the red blood cell, and that CO2 binds with, and hemoglobin has the symbol HB, and it forms HbCO2, and that, again, I'll just write the name, is carbaminohaemoglobin. And as I said, this process is reversed in the lungs. Now that accounts for about roughly or so 10% of the carbon dioxide that we've produced. So that's roughly about 10%. So you can see 5% dissolves in the plasma, 10% dissolves or passes rather into the red blood cell and binds with hemoglobin to form carbaminohaemoglobin. So we now have another 85% of carbon dioxide that is produced to account for. And the 85% is transported as what's called hydrogen carbonate ions. Now the carbon dioxide actually dissolves in water to form what's called carbonic acid. and we use a catalyst, carbonic anhydrase, to do that, and then that would eventually dissociate to form hydrogen carbonate ions. Let's take this step by step and I'll explain what's going on. So let's take this CO2 here. Now this would be, again, within the red blood cell, but I'll do a little arrow there because technically I don't have any more room in the picture of the red blood cell. I'm just going to draw it a bit below it. So what we actually have is carbon dioxide binding with water, and it forms what's called H2CO3. Now that is carbonic acid, so we'll just put that in there. That's carbonic acid, and we're going to use a catalyst to do so. And this catalyst is really important. Exam questions often refer to this catalyst, so we'll put the name in there of the catalyst. And the catalyst that we're using is one called carbonic anhydrase. Notice it ends in ASE. Surefire sign of an enzyme. So we've got a catalyst there, carbonic anhydrase, catalysing the reaction between carbon dioxide and water, and we're going to produce carbonic acid. Now that carbonic acid then dissociates, and it dissociates into HCO3 minus, they're hydrogen carbonate ions, and we also get H plus hydrogen ions. Now remember, this is within the red blood cell. This is within the red blood cell. So we've had that carbonic acid dissociate, and we've produced some hydrogen carbonate ions and hydrogen ions. Now those hydrogen carbonate ions, those HCO3 minus ions, diffuse out of the cell, and Cl minus, so chloride or chlorine ions, diffuse into the cell to balance that charge, and it's that's known as the chloride shift. So if we were to just take this HCO3 minus ion here, and I were to just write it in the red blood cell as if we've formed that, so there's the HCO3 minus. Essentially what we have is the HCO3 minus leaving, diffusing out the cell, and Cl minus chloride ions coming in. And that there is known as the chloride shift. Now let's think about what those H plus ions are going to do. Because down here, I said we get some H plus ions, so let's think about those. Those H plus ions will bind with hemoglobin. They bind with hemoglobin to form what's called hemoglobinic acid. And these ions, ultimately, you could say therefore, compete with oxygen for the hemoglobin. Ideally, hemoglobin binds with oxygen, it forms oxyhemoglobin, but what we're saying now is that these H plus ions are actually going to compete with the oxygen and bind to that hemoglobin instead, forming hemoglobinic acid. So if we were to if we shrink the screen just a small amount here, just so I can continue writing beneath. So those H plus ions, those will bind with hemoglobin, so it looks a bit like this. HB, symbol for hemoglobin, plus H plus, will go to produce what's called HHB, and that is known as, and I'll write it here again, hemoglobinic acid. So hemoglobinic acid. And I said they're going to compete with oxygen for the hemoglobin. Now, the increase in hydrogen ions causes the pH level to fall. So the hemoglobin actually acts as a buffer by attracting them, preventing damage. Plasma proteins in the blood also have this kind of buffering effect. So now we've considered the three mechanisms of carbon dioxide transport, the 5% dissolved in the plasma, and the 10% binding with hemoglobin to form carbaminohaemoglobin, and the remaining 85% forming carbonic acid and then dissociating into H plus and HCO3 minus ions, we now need to consider something known as the Boer Shift and really put all this into a context. So again, we'll just shrink the size of the screen a little bit more. Let's put this into a real context. Let's say we have just a very quick sketched graph at the bottom here, and on the x-axis we have, let's put the PO2. Now that symbol, the small p, means partial pressure of oxygen, and that's going to be in kilopascals. And up the y-axis we'll have percentage saturation of hemoglobin with oxygen. So we'll just put this as percentage sat for the moment. Now usually when you look at the oxygen dissociation curve, it looks a bit like this. It has a characteristic S shape. Now what we find is that when the carbon dioxide level increases, so when we're in a carbon dioxide rich environment, this curve actually appears to shift to the right. So when the CO2 level increases, this whole curve seems to shift to the right. And let's think about what that means. In a CO2 rich environment, so we're talking about maybe the respiring tissue, more carbonic acid is formed. Now that ultimately would mean that more H plus ions are released. So there's more competition for hemoglobin. Now as a result, more oxygen would dissociate from oxyhemoglobin. So that's why the oxygen dissociation curve shifts to the right, as the body now requires essentially a higher partial pressure of oxygen to effectively saturate that hemoglobin. And it's this that's known as the Boer Shift. So this phenomenon is known as the Boer Shift. So just to reiterate what I've said there, CO2 rich environment, more carbonic acid ultimately forms. When that dissociates, you get more hydrogen carbonate and more H plus ions. So more hydrogen ions are released. They're going to compete with oxygen for hemoglobin. So ultimately what you're going to end up with is more oxygen dissociating from oxyhemoglobin. So to reflect that, the oxygen dissociation curve would shift to the right. So because it shifted to the right, it means for any given partial pressure of oxygen, the saturation of hemoglobin is less. So when it's saturating the hemoglobin less, the oxygen is actually being released a lot easier. It's coming off a lot more. So more carbon dioxide ultimately would release more oxygen. And that is known as the Boer Shift. Okay, hope all that helps.